Okay, let's unpack this. Have you ever wondered what truly powers your body when you haven't eaten for hours? Or maybe during that last grueling mile of a run? It's not always glucose. Today, we're taking a deep dive into the truly fascinating world of how your body expertly harnesses energy from fat.
That's right. Our mission today is really to guide you step by step through the core concepts, the biochemical pathways, and, even some compelling clinical examples surrounding the oxidation of fatty acids and ketone bodies. We're drawing our insights from Chapter 30 of Marks's Basic Medical Biochemistry. Think of fatty acids as your body's incredibly dense, sort of long-term energy storage. And ketone bodies, well, they're those clever alternative fuels that keep things running smoothly when glucose becomes scarce. This deep dive will hopefully give you a metabolic shortcut to being genuinely well-informed with plenty of aha moments about your own internal fuel system.
So, to kick us off, where do these power-packed fatty acids actually come from, and why are they such a critical part of our energy strategy?
Well, fatty acids are, without a doubt, a primary fuel source for humans. They become the major energy provider for hardworking tissues like your heart muscle, skeletal muscle, and the liver, especially during periods between meals, say an overnight fast, or when energy demand spikes, like during intense exercise.
Okay.
And your liver also plays this special, almost visionary role here. It can convert some of these fatty acids into what we call ketone bodies, specifically acetoacetate and β-hydroxybutyrate.
Ah, the ketone bodies.
Exactly. And these then serve as alternative fuels for many other tissues, even the brain, which is really important, particularly during prolonged fasting.
It sounds like our body is constantly looking for the most efficient fuel for the job at hand. And we also classify fatty acids by their chain length, right? That's important because it dictates how they'll be metabolized.
Absolutely. Yeah, we have very long chain ones, long chain, medium chain, and short chain fatty acids. The primary ones we tend to use are typically long chain fatty acids like palmitate, oleate, and stearate.
Okay, the common ones.
Right. These are abundant in our diet, and our liver can also make them. The body then stores these mainly as triacylglycerols within adipose tissue. That's our fat reserves.
Right.
Then, when fuel is needed, this precise symphony of hormonal signals kicks in. You get a decrease in insulin and an increase in hormones like epinephrine, norepinephrine, or glucagon.
The get up and go signals?
Kinda, yeah. This hormonal shift triggers the release of these stored fatty acids from the adipose tissue. This is exactly what's happening for Otto S, the marathon runner case study, as his muscles demand more and more energy during those long runs.
It's incredible how our body adapts on the fly. Like you mentioned, after an overnight fast, a huge chunk of our energy comes from fat.
Yeah, a significant portion, we're talking about 60% to 70% of our energy, actually comes from oxidizing these fatty acids, not glucose.
Wow.
And this brings us to a case like Lola B, who experienced symptoms only when she fasted for more than 8 hours. That was a clear hint that something might be, amiss with this essential fatty acid metabolism.
Yeah, like her body was running on fumes when its usual fatty acid engine wasn't firing properly.
Now, before they can actually be used for fuel, these fatty acids have quite a journey to take. They're naturally hydrophobic.
Meaning they don't mix well with water.
Exactly. And at high concentrations, they can even be a bit toxic. So they need reliable escorts, basically, through the bloodstream and into the cells.
Like VIP passengers needing a secure ride. So in the blood, what's carrying these long-chain fatty acids around safely?
In the blood, they bind tightly to albumin. That's the most abundant protein in our serum. You can picture these fatty acids sort of nestled snugly into a specialized hydrophobic pocket on the albumin molecule, protected and transported safely to their cellular destinations.
Okay.
Once they arrive at a cell, they get inside either by simple diffusion or through dedicated transporter proteins on the cell surface. This keeps the concentration of free, potentially harmful fatty acids inside the cell extremely low.
So they get inside, but they're not quite ready for action yet, right? What's the next step? There's an activation step.
Precisely. Next comes activation. They have to be converted into acyl coenzyme A, or acyl-CoA derivatives. This process uses enzymes called acyl-CoA synthetases, or thiokinases, and it requires an initial energy investment. It actually costs ATP.
Ah, you got to spend energy to make energy.
Kind of. It cleaves 2 high-energy phosphate bonds from ATP. It's like a down payment to get the energy engine officially running. ATP gets broken down to AMP and pyrophosphate, which really drives this crucial reaction forward.
And I understand where this activation happens can actually vary depending on the fatty acid. It's not always in the same place.
That's right. There are different types of these acyl-CoA synthetases, sort of strategically located. For long-chain fatty acids, you'll find them in the endoplasmic reticulum, on the outer mitochondrial membranes, or even in proximal membranes. But for medium-chain ones, the activation can happen right inside the mitochondrial matrix, especially in liver and kidney cells. It's a bit more direct for them.
Okay, so once they're activated, acyl-CoAs, then what? Where do they go?
Well, their fate can vary. Some might be used to build new triacylglycerols or phospholipids, for cell structures. But for our energy story today, the activated long-chain fatty acyl-CoAs have a very specific destination. They need to get inside the mitochondrial matrix. That's where the main energy extraction happens.
Okay, into the powerhouse.
Exactly. And this is where a specialized carrier system, known as the carnitine shuttle, becomes absolutely essential. Carnitine is the key carrier molecule here.
Describe this shuttle for us. It sounds like a really precise operation to get past those mitochondrial gatekeepers. How does it work?
It is, yeah. It's a marvel of cellular logistics. On the outer mitochondrial membrane, there's an enzyme called carnitine palmitoyltransferase, CPTI for short, sometimes called CPTII. CPTI takes the fatty acyl group from CoA and skillfully transfers it to carnitine, forming an acyl-carnitine ester. Think of it like a molecular bridge.
Okay, handing it off.
Right. This acyl-carnitine then crosses the inner mitochondrial membrane with the help of a specific transporter protein, a translocase. Once safely inside the mitochondrial matrix, a second enzyme, carnitine palmitoyltransferase, the second CPTI, or CPTI, does the reverse. It transfers the fatty acyl group back to CoA.
Ah, so it reconstitutes the original activated fatty acid inside.
Precisely. This regenerates the fatty acyl-CoA, now ready for oxidation, and it simultaneously releases the carnitine. The carnitine then cycles back out via the translocase to pick up another fatty acyl group. It's an incredibly efficient cyclical system.
So carnitine is clearly critical for getting those long-chain fatty acids to their final energy destination. Otto S, our marathon runner, you mentioned he even included carnitine in his supplements, but normally we make enough.
Yeah, generally our bodies synthesize enough of it from the amino acid lysine, and we also get it from our diet, especially meat. So it's typically only in specific deficiencies, maybe genetic ones or certain medical conditions, that it becomes a real problem.
And when it is a problem?
Well, the consequences can be severe. Inherited defects in this carnitine transport system, like a CPTII deficiency, can cause some serious issues. Things like recurrent episodes of muscle pain, weakness, and what we call hypoketotic hypoglycemia.
Hypoketotic hypoglycemia. Break that down. Low sugar and low ketones.
Exactly. Not only dangerously low blood glucose, but also, crucially, the body isn't producing enough of those alternative ketone fuels to compensate. The body is left in a critical energy deficit. These patients often accumulate lipid deposits in their muscles, and you'll see elevated creatine phosphokinase and long-chain acyl-carnitines in their blood, simply because the fatty acids can't get into the mitochondria to be burned.
Okay, so once those fatty acyl-CoAs are finally inside the mitochondrial matrix, safely past the carnitine shuttle, the real work of dismantling them for energy begins. What's this fundamental process, beta-oxidation, all about? How does it work?
Beta-oxidation is, well, it's an ingenious process. It's not really a cycle, it's more like a spiral. It repeatedly chops off 2 carbon units in the form of acetyl-CoA from the carboxyl end of the fatty acyl group.
Like a molecular zipper, 2 carbons at a time.
That's a great analogy. Yeah, it progressively shortens the fatty acid chain 2 carbons at a time. Each turn of this spiral prepares the fatty acid for the next cut, meticulously extracting energy along the way.
And this spiral involves a series of distinct steps, doesn't it? Can you walk us through those stages?
Exactly. It's a very precise 4-step process for each turn. First, an acyl-CoA dehydrogenase enzyme creates a double bond between the alpha and beta carbons. This is an oxidation step, and it transfers electrons to FAD, producing FAD2H.
Okay, first oxidation.
Second, an oil-CoA hydratase adds a molecule of water across that new double bond. This puts a hydroxyl group on the beta carbon.
Hydration, adding water. Makes sense.
Third, a hydroxyl-CoA dehydrogenase enzyme oxidizes that hydroxyl group to a ketone group. This generates NADH by transferring electrons to NAD plus lead.
Second oxidation.
And finally, fourth, a beta-ketothiolase enzyme cleaves the bond right between the alpha and beta carbons. This precisely releases 1 molecule of acetyl-CoA and leaves behind a new fatty acyl-CoA, which is now 2 carbons shorter than the original.
And that shortened one goes back to step 1.
Exactly. It re-enters the spiral, repeating the whole 4-step process until the entire fatty acid chain is broken down into acetyl-CoA units.
That's a lot of intricate steps for each term, but it's all about generating energy. How much ATP are we actually talking about from this whole breakdown? It sounds like a lot.
Oh, it is a lot. This is where the sheer efficiency of fat as fuel really shines. Each FAD2H produced in the spiral eventually generates about 1.5 ATP via the electron transport chain, and each NADH generates about 2.5 ATP.
Okay.
Then the acetyl-CoA molecules, which are the main output, they enter the TCA cycle, the citric acid cycle, yielding about 10 HEP each.
Right.
So let's take palmitate, a common 16-carbon fatty acid. It needs 7 turns of the oxidation spiral to break down completely. This produces 7 FAD2H, 7 NADH, and 8 molecules of acetyl-CoA.
Okay, let's do the math. So roughly, 7 times 1.5 ATP plus 7 times 2.5 ATP plus 8 times 10 ATP, that's 10.5 plus 17.5 plus 80, which gives us 108 ATP.
108 ATP.
Oh, wait, remember that initial activation step? It cost us 2 ATP equivalents.
Ah, right, the investment.
So the net yield is an impressive 106 ATP. The key takeaway here isn't just the number, but the sheer energy density. One molecule of palmitate gives us a staggering 106 ATP. Fat is incredibly energy-rich. That's why it's our body's primary long-duration fuel. Truly an impressive energy payout.
That's truly a huge amount of energy. But what if the fatty acid isn't a perfect, straight, even-numbered chain? You mentioned palmitate, which is C16. What about others?
Excellent question, because yeah, about half of our dietary fatty acids are unsaturated. They have those cis double bonds like oleate or linoleate. Beta-oxidation normally creates a trans double bond in step 1, so special auxiliary enzymes are needed to handle these cis bonds.
So the spiral needs a little help sometimes. Exactly. For example, with linoleate, you need enzymes called isomerases to basically move and reconfigure the double bonds into the correct position and configuration for the beta-oxidation enzymes. Sometimes, you even need an NADPH-dependent reductase enzyme to deal with conjugated double bonds, converting them into a single trans double bond that the spiral can handle.
Clever workarounds.
And what about fatty acids with an odd number of carbons? What happens when the spiral gets down to the last 3 carbons? They can't top off 2 then.
Right. They get broken down turn by turn, just like the others, until the very last spiral leaves a 3-carbon molecule called propionyl coenzyme A. This propionyl-CoA then undergoes a special multi-step pathway. It gets carboxylated to methylmalonyl-CoA and then finally converted into succinyl-CoA.
Succinyl-CoA, that's a TCA cycle intermediate.
Precisely. This last step is incredibly significant because it requires vitamin B12, and it feeds directly into the TCA cycle. This means that this small 3-carbon portion of an odd-chain fatty acid can actually contribute to glucose synthesis through gluconeogenesis, a crucial detail. This is where we start seeing those serious clinical implications again, like with Lola B's case. You mentioned her specialist suspected a fatty acid metabolism disorder. Why? What were the specific clues?
Lola B's clinical picture was quite specific, yeah. Her urine showed elevated levels of partially oxidized medium-chain fatty acids, things like octanoic acid, and also unusually high levels of dicarboxylic acids, along with medium-chain acylcarnitines.
Okay, a specific pattern.
Yes, this unique profile strongly pointed towards a medium-chain acyl-CoA dehydrogenase deficiency, or MCAD deficiency.
Right.
In this relatively common inherited disorder, the long-chain fatty acids get broken down okay initially, but then they hit a roadblock at the medium-chain length because the MCAD enzyme isn't working properly.
So they get stuck mid-process.
Exactly. The body tries to compensate, but it just can't fully oxidize the fat. This leads back to that hypoketotic hypoglycemia we discussed, low blood sugar, but critically, also low ketone bodies, leaving the body without sufficient alternative fuel during fasting. We see similar energy crises in cases like Jamaican vomiting sickness, which is caused by a toxin that specifically inhibits acyl-CoA dehydrogenases, forcing the body to over-rely on glucose and leading to severe, potentially fatal hypoglycemia.
So if that main mitochondrial beta-oxidation pathway, which sounds so central, hits a snag like an MCAD deficiency, are there backup plans? Does the body have other ways to handle problematic fatty acids?
Absolutely. Our bodies are incredibly resilient and have what we call alternative oxidation pathways. These are mainly located in peroxisomes and also in the endoplasmic reticulum. Think of them as auxiliary processing plants, stepping in when needed.
Okay, backup systems.
Right. These are crucial for handling things like very long-chain fatty acids, those with 20 or more carbons, which don't get handled well initially by the mitochondria. They also deal with branched-chain fatty acids, or even certain foreign compounds, xenobiotics, that might look like fatty acids to the cell.
So these auxiliary plants pick up where the main system struggles, like for those really long chains.
Precisely. In peroxisomal oxidation, for instance, very long-chain fatty acids are partially oxidized inside peroxisomes. They get shortened down to about 4 to 6 carbons. An interesting difference here. The first step in peroxisomal oxidation uniquely produces hydrogen peroxide, H2O2, directly, using an oxidase enzyme, instead of capturing the energy as FADH₂, like in mitochondria.
Hydrogen peroxide, so that needs to be dealt with too.
Yes, peroxisomes have catalase to break that down safely. But the key thing is the shortened fatty acid products from the peroxisome are then efficiently transferred to the mitochondria for complete oxidation there.
Okay, peroxisomes do the initial shortening. And what about those tricky branch chain fatty acids like phthalic acid from green vegetables?
Right, those methyl branches can block normal beta-oxidation. So for those, peroxisomes employ a specialized pathway called oxidation first. Phthalic acid is a good example, a multimethylated C20 fatty acid. Alpha-oxidation, using enzymes like propionyl-CoA hydroxylase, cleverly removes the first carbon as CO2.
Removes the first carbon.
Yeah. It's ingenious because this strategically shifts the positions of those methyl groups so they no longer block the subsequent beta-oxidation steps. Then beta-oxidation can proceed, and again, the shortened products are eventually sent to the mitochondria for final energy extraction.
And this is another area with serious clinical consequences if things go wrong, right? Like Zellweger syndrome or Refsum disease.
Exactly. In Zellweger syndrome, the peroxisomes themselves are defective, so you get a dangerous buildup of very long-chain fatty acids that can't be processed. Refsum disease, on the other hand, is a specific deficiency in the oxidation pathway for phthalic acid, causing it to accumulate. Both conditions really highlight how critical these alternative pathways are.
So we have peroxisomes handling the really long and branched chains. Are there any other alternative pathways?
Yes, there's also oxidation, that's ω-oxidation, which takes place in the endoplasmic reticulum. This pathway works differently again. Instead of breaking down the fatty acid from the carboxyl end, the alpha-beta end, it oxidizes the terminal methyl group, the very last carbon at the other end of the chain, the omega carbon.
Attacks it from the other end?
Kind of, yeah. It converts that omega carbon first to an alcohol, then to a carboxylic acid group. This process produces dicarboxylic acids, which now have carboxyl groups, dicar-CO, at both ends of the molecule.
Ah, so they're different.
Yes, and this makes them significantly more water-soluble. They can then potentially undergo beta-oxidation from both ends, and importantly, they're easily excreted in the urine.
And this connects back to Lola B again, doesn't it? You mentioned dicarboxylic acids in her urine earlier.
It does, absolutely. Lola B's urine showing those elevated C6 and C8 dicarboxylic acids, adipic and seborrheic acids, along with octanoyl glycine. It strongly suggests her body was actively using oxidation as a compensatory mechanism. It was trying to deal with those medium-chain fatty acids that were building up because her MCAT enzyme was defective.
So the body was shunting them to this alternative pathway to try and get rid of them.
Exactly. While these alternative pathways, omega and alpha oxidation, are usually minor players in overall fat burning, they become absolutely vital when mitochondrial beta oxidation is compromised. And their rate seems to be primarily regulated just by the availability of their substrates, not by complex feedback loops like the main pathway.
We've now really dug into how fatty acids are oxidized. Let's pivot to their fascinating offspring, as you called them, ketone bodies. These ingenious alternative fuels the liver cooks up.
Right. During fasting, or any state where fatty acids become abundant, the liver truly becomes a major producer of ketone bodies. And it's important to stress, these aren't just metabolic waste products. They're critical energy molecules for many other tissues, especially when glucose is running low. The two main ketone bodies we focus on are acetoacetate and β-hydroxybutyrate. There's also a small, volatile amount of acetone produced.
So how exactly does the liver go about making them? What's the process?
Ketone body synthesis, or ketogenesis, occurs exclusively in the mitochondrial matrix of liver cells. When acetyl-CoA levels get really high from lots of fatty acid oxidation, it signals an abundance of fuel building up. Two molecules of acetyl-CoA basically reverse the last step of beta-oxidation to form aceto-acetyl-CoA.
Okay.
This aceto-acetyl-CoA then reacts with another acetyl-CoA molecule to produce a key intermediate called HMG-CoA. This reaction is catalyzed by HMG-CoA synthesis.
HMG-CoA, that sounds familiar from cholesterol synthesis too.
It does. There's a cytosolic version involved in cholesterol synthesis, but this mitochondrial HMG-CoA synthase is dedicated to ketone body production. Finally, an enzyme called HMG-CoA lyase cleaves this mitochondrial HMG-CoA to yield one molecule of acetyl-CoA and one molecule of acetoacetate. That's our first ketone
The
bio.
acetoacetate and the other one, beta-hydroxybutyrate.
Right. Acetoacetate can then either directly enter the blood, as is, or it can be reduced to NADH-hydroxybutyrate. This is a reversible reaction, and the balance between them, the ratio, is largely determined by the mitochondrial NADH-NAD plus ratio. Typically, it favors hydroxybutyrate, maybe around 3.1 or so.
Okay, so the redox state matters.
It does. And acetoacetate can also spontaneously decarboxylate, just break down on its own, forming CO2 and that volatile acetone. That's what you might sometimes smell in the breath in severe ketosis, often described as a fruity odor.
It's fascinating how the liver produces these alternative fuels, but here's a key question. Can the liver itself actually use them for energy, or is it just exporting them?
Surprisingly, and this is a really key detail, the liver largely synthesizes ketone bodies for other tissues. It's quite altruistic in this way. The liver itself lacks sufficient amounts of a crucial enzyme called succinyl-CoA acetoacetate-CoA transferase, sometimes called deophorase. This enzyme is absolutely required to activate acetoacetate and convert it back to acetoacetyl-CoA, so it can be burned for energy. So the liver makes the fuel, but can't really use it itself.
That's a critical piece of the puzzle, so it ships them out. How do these other tissues, like our muscles and even the brain, actually put ketone bodies to work then?
Well, most other tissues, including skeletal muscle, the heart, parts of the kidney, the intestinal lining, and crucially the brain, are fully capable of oxidizing ketone bodies. They actively transport ketone bodies from the blood into their own mitochondria. Once inside, bihydroxybutyrate is first oxidized back to acetoacetate, and this step actually generates a molecule of an ADH bonus energy.
Oh, interesting.
Then, acetoacetate is activated back to acetyl-CoA by that specific transferase enzyme, the one the liver is missing. It uses succinyl-CoA from the TCA cycle to do this. This acetyl-CoA is then quickly cleaved by thiolase into two molecules of acetyl-CoA.
And those acetyl-CoAs just feed right into the TCA cycle.
Directly into the TCA cycle for significant ATP production. For example, one molecule of base-hydroxybutyrate can yield approximately 21.5 ATP. It's a substantial energy source, especially for the brain during prolonged fasting.
This ability for the brain to use ketone bodies is precisely why we hear about ketogenic diets sometimes being used therapeutically, isn't it?
Exactly. Ketogenic diets, which are very high fat and low carbohydrate, force the body into a state of nutritional ketosis. They're used, for example, to help reduce epileptic seizures in some children, particularly those refractory to medication, and also to treat specific metabolic conditions like pyruvate dehydrogenase, PDH deficiency. They effectively provide that vital alternative fuel for the brain when glucose use is limited. We also see certain amino acids like leucine and isoleucine, often called ketogenic amino acids, because their breakdown products can directly contribute to ketone body synthesis in the liver.
And of course, we can't really talk about ketone bodies without thinking of the dangerous side. Like in Dianne A's case, experiencing diabetic ketoacidosis, DKA. What's the dangerous cascade happening there?
Right. DKA is a severe and potentially life-threatening complication, most often seen in type 1 diabetes, but sometimes in type 2 as well. What happens is an acute severe insulin deficiency, often coupled with an excess of counterregulatory hormones like glucagon. This hormonal imbalance causes the rapid and uncontrolled mobilization of fatty acids from adipose tissue lipolysis goes into overdrive.
A flood of fatty acids hits the liver?
Exactly. The liver, receiving this huge influx of fatty acids, dramatically ramps up ketone body synthesis, essentially making far, far more than the body can use. Compounding the problem, the insulin deficiency also severely impairs the ability of peripheral tissues to take up and use both glucose and ketones. So you get this buildup, leading to dangerously high levels of ketone bodies in the blood, sometimes 8 to 15 millimolar or even higher, compared to normal fasting levels of maybe 0.2 to 2 millimeter.
And these ketone bodies are acidic.
Yes. Acetoacetate and phenhydroxybutyrate are acidic. These incredibly high levels overwhelm the blood's natural bicarbonate buffering system. This leads to a drop in blood pH, a metabolic acidosis. The body tries to compensate by breathing rapidly and deeply, that characteristic Kussmaul breathing, trying to blow off CO2. But if it's not treated quickly with insulin and fluids, it can rapidly progress to dehydration, electrolyte imbalance, coma, and even death. It really underscores the critical need for balance in fuel metabolism.
It's abundantly clear that fatty acids and ketone bodies are absolutely crucial fuels. But how does the body decide when to use them and, maybe more importantly, how much to use? How is this whole grand metabolic symphony orchestrated?
It's truly an intricate dance of fuel homeostasis, a real masterpiece of biological regulation. Basically, these fuels, fatty acids, and ketones are prioritized and brought to the forefront whenever the levels in the blood are elevated. This happens during fasting, starvation, prolonged or intense exercise, or when someone follows a very high-fat, low-carbohydrate diet.
So availability drives usage to some extent.
To a large extent, yes. And the hormonal environment is key. That decreased insulin to glucagon ratio, which signifies a fasting or low energy state, is the main trigger. It triggers lipolysis in adipose tissue, increasing fatty acid release. And then in the liver, this same hormonal signal promotes ketone body synthesis.
So the hormones are truly like the conductor of this metabolic orchestra, telling different sections like fat tissue and the liver when to play their parts.
Precisely. And the regulation of beta-oxidation itself, the burning of fatty acids, is tightly linked to the cell's immediate energy demands. Probably the most critical regulatory point for getting long-chain fatty acids into the mitochondria is that first enzyme of the carnitine shuttle, CPTI.
CPTI. And how is that regulated?
It's exquisitely inhibited by a molecule called malonyl coenzyme A, or malonyl-CoAs.
Malonyl-CoA, you mentioned that earlier. That sounds familiar from fatty acid synthesis, right?
It is indeed. Mol-CoA is the key building block, synthesized by the enzyme acyl-CoA carboxylase, ACC. And this connection reveals a truly brilliant regulatory strategy. Think about it. When the liver is actively synthesizing new fatty acids, meaning it's in a fed state, signaled by high insulin ACC is active, and malonyl-CoA levels are high.
Okay.
This high malonyl-CoA then inhibits CPTI. It effectively closes the gate to the mitochondria for fatty acid entry.
So it prevents the newly made fats from immediately being burned.
Exactly. It's a neat way to ensure that newly synthesized fats are directed towards storage as triacylglycerols or export, not futilely burned right away. It prevents the body from working against itself.
That makes perfect sense. And conversely, during fasting or exercise.
Conversely, during demanding periods like intense exercise or fasting, cellular AMP levels rise, signaling low energy charge. This rise in AMP activates AMP-activated protein kinase, or AMPK. AMPK then phosphorylates and inactivates acetyl-CoA carboxylase, ACC.
So it shuts down fat synthesis.
Right, which means malonyl-CoA levels drop. When malonyl-CoA levels fall, the inhibition on CPTI is relieved. The gate opens, and fatty acid oxidation ramps up to generate ATP and restore energy balance. This is exactly what's happening in Otto S's muscles as he runs his marathon, masterfully switching to fat burning.
So it's a reciprocal regulation. High energy fed state blocks burning. Low energy fasting state promotes burning. Very elegant. Now, if fatty acids and ketone bodies are plentiful, can they actually tell the body to use less glucose, almost like preserving the glucose?
Yes, that's exactly right. It's a profound glucose-sparing effect, particularly important during prolonged fasting or starvation. High rates of fatty acid oxidation generate significant amounts of NADH and ATP within the mitochondria. These energy signals, along with the high levels of acetyl-CoA produced, then feed back to inhibit key enzymes in glucose breakdown, or glycolysis.
Which enzymes get inhibited?
Key regulatory points like pyruvate dehydrogenase, PDH, which links glycolysis to the TCA cycle, and phosphofructokinase 1, PFK1, a major control point within glycolysis itself. Citrate produced from acyl-CoA also inhibits PFK1. So this effectively slows down glucose uptake and glycolysis in tissues like muscle, reserving that precious glucose for tissues that absolutely depend on it.
Like red blood cells.
Exactly. Red blood cells lack mitochondria, so they can only use glucose. And the brain, while it can adapt to ketones, still needs some glucose. So this sparing effect is vital.
That's a remarkable amount of intricate control already, but I think you mentioned something earlier, or maybe I read recently, about acetylation playing a role too. This sounds like a newer layer of regulation being discovered.
Indeed. It's a really exciting area. Recent research highlights acetylation, specifically of proteins, as another fascinating and complex layer of metabolic regulation. It shows just how much more we're still learning. Similar to how histone acetylation regulates gene expression, certain mitochondrial enzymes involved directly in fatty acid oxidation can themselves be acetylated.
So adding an acetyl group to the enzyme itself.
Yes. For example, very long-chain acyl-CoA dehydrogenase, or VLCAD, can be acetylated on certain lysine residues, and this acetylation seems to inhibit its activity. Now enter the sirtuins. This is a family of NAD plus independent protein deacetylases. SIRT3 is a key one located in the mitochondria.
Okay, so sirtuins remove acetyl groups.
Right. And importantly, sirtuins like SIRT3 are often upregulated during fasting or caloric restriction times when we need to burn fat. So SORT3 deacylates enzymes like VLCAD, removing that inhibitory acetyl group and thereby activating the enzyme.
Ah, so fasting boosts RT3, which activates fat-burning enzymes by deacetylating them.
Exactly. It leads to increased fatty acid oxidation, particularly in the liver. You can imagine the sirtuin reaction removing an acetate group, almost like flipping a switch to turn the enzyme back on or boost its activity. This adds an incredible depth of complexity to how our bodies fine-tune energy metabolism, allowing for incredibly precise responses to fuel availability and cellular energy status. It's not just about hormones and simple feedback anymore. Hashtag tag tag outro.
Wow, what an intricate system. We've really journeyed through the sophisticated world of fatty acid and ketone body oxidation today, from their initial release and that careful activation process, through their detailed breakdown in beta-oxidation and those alternate pathways, and finally, the really ingenious ways our body regulates it all. We have seen firsthand how absolutely essential these fuels are, especially during fasting or intense activity, and how disruptions, like MCAD deficiency or diabetic ketoacidosis, can have truly profound clinical consequences.
You definitely now have a clear understanding, I hope, of your body's remarkable ability to switch and adapt its fuel sources.
Yeah.
It really provides a well-informed perspective on metabolism. And thinking about the sheer efficiency, the redundancy built into these systems and these newer discoveries, like acetylation's role in regulation,
Hmm.
it really raises an important question, doesn't it?
It does. So here's something to think about. What stands out to you from all this? As we continue to uncover these incredibly intricate layers of metabolic control, what other hidden mechanisms might be shaping our health and disease? And maybe more importantly, how might future therapies leverage these detailed insights to target metabolic disorders like obesity, diabetes, or even those genetic conditions more effectively? Thank you so much for joining us on this deep dive today. We hope you're leaving with a much deeper appreciation for the complex biochemical symphony that keeps you powered every single day. From the entire Last Minute Lecture team, thank you for listening.